So welcome to week 3 in this Coursera course on epigenetic control. You'll remember in the first 2 weeks we've run through chromatin modifications and the various epigenetic marks, how chromatin is packaged and how this all relates to transcriptional control, so whether the gene is likely to be silenced or expressed. And now in the third week what we want to consider is dosage compensation. We've spoken a little bit about dosage compensation in mammals, about X inactivation on a few occasions as a means to explain some of the things that we've been speaking about so far. But now we're going to delve into more detail. The reason we're doing this is because not only is it a fascinating process where a whole X chromosome, about 1,000 genes, goes unused and densely packaged in the nucleus. Also, what we learn about dosage compensation tends to be true for epigenetic control throughout the rest of the genome, and so it makes a really good model system to think about when we want to try and put all of these epigenetic marks together that we've spoken about in the previous 2 weeks to try and understand at a molecular level how epigenetic control actually functions. So we'll start out with a bit of a historical view of X inactivation. We'll go through the various stages that exist in X inactivation in mammals and the molecular mechanisms about how these work, at least what's known about them so far. We're then going to take a very brief foray into thinking about how dosage compensation exists in lower organisms, in flies and worms. And finally, we'll spend a little bit more time thinking about a few other things that we've learnt about from the fly. And in fact, much of what we know about epigenetic control in mammals has come from work that's been done in flies and then been translated into the mammalian system. So let's think about X inactivation in mammals then. Well, it was way back in 1949 when Barr and Bertram discovered that when they were looking at the nuclei of neurons from cats, that in the male nuclei compared with the female nuclei, they didn't observe this dense heterochromatic body. In the female nuclei, what they saw was a region of densely packed DNA, of heterochromatin, that was found at the nuclear periphery. And so you can see in the image that's shown here on the right of the slide that here, indicated by the arrow, that there is a region that is stained more densely, this heterochromatic region, and this is now known as the Barr body. So it was then in 1959 that Setsuko Ono discovered that this wasn't just any old region of heterochromatin, but in fact this region, which was exclusively found in female cells, was indeed an X chromosome. So moving on from that in 1959, this discovery in 1959, in 1961 Mary Lyon proposed that this X chromosome was inactivated in order to perform dosage compensation for male cells which had only one X chromosome in comparison with female cells that had two. And really, one of the hallmarks of Mary Lyon's hypothesis was that this choice of which X chromosome would be inactivated was random. So you remember that we've spoken about on other occasions that there is random X inactivation, and either the paternally inherited or the maternally inherited X chromosome could be chosen to be inactivated, and that this choice is then heritable for the life of that cell and all of its progeny. So we'll think about this again in a couple of slides' time and go through each of these features that Mary Lyon proposed. But it's nice to note that 50 years after Mary Lyon first made her hypothesis, so Lyon's hypothesis was changed to Lyon's law. So now this is taken as law, that this is indeed what happens in the mammalian cell. So you can also see another image, which I'll show you, which is a further image related to this first one, that on the lower panel we're staining just DNA, and you can see a denser region of DNA at the nuclear periphery indicated with the arrow, the Barr body. But in the upper panel, what we're looking at now is X chromosome DNA, and this is painted with a chromosome paint. And so the one that's densely stained, there are two regions, one that is densely staining and one that is less densely staining, and we actually don't see any dense staining here at all where there is this second X chromosome. And so these denote the inactive X chromosome, which is densely staining and at the nuclear periphery, this Barr body, while the open chromatin, so therefore this not particularly well-stained region, is the active X chromosome. Another way to see the Barr body is shown in this slide, where you can see on the left we're staining the DNA in blue with a DNA intercalating dye. And again, you can see indicated with the arrow the Barr body, this region that's more densely staining at the nuclear periphery, which is only found in the female cells. However, You can then see in the right panel, we're staining for a histone variant that I mentioned in previous lectures called macro H2A. And macro H2A is known to be accumulated on the inactive X chromosome. And so there's a co-localization of this densely DAPI staining or dense DNA, the heterochromatin, this bar body, and macro H2A, as you can see, is indicated by the arrow in this immunofluorescence image. So it's nice to think about how it was that Mary Lyon and also Tsuzuka Ono were able to come up with their theory, Mary Lyon's theory of X inactivation and Tsuzuka Ono's really amazing discoveries about the X chromosome. So one of the ways they were able to propose this was because they were looked in karyotypically abnormal cells, so cells that didn't contain normal numbers of chromosomes. So let's think about the two normal cases first. We know that in female mammals you have 2 X chromosomes and 44 autosomes. This is in a human. This is a normal diploid female. In normal males you have 1 X chromosome, 1 Y chromosome, and still 44 autosomes. But then there are different cases where this is not the case. We have an abnormal number of sex chromosomes, or X or Y chromosomes. So the first case is trisomy X. And then, as it sounds like, you have 3 X chromosomes. These patients have 3 X chromosomes, but still the normal number of autosomes. Klinefelter's patients, who are males, they have an X and a Y like your average male would, but they have an additional X chromosome. So 2 X chromosomes and a Y chromosome, plus their normal 44 autosomes. And finally, an additional type that was studied were tetraploid female cells. These aren't from patients, but what they found was that in the dish, if you had cells that instead of having a diploid content, but instead had doubled all of that, so now they had 4 X chromosomes and 88 autosomes, by looking at each of these and counting the number of bar bodies, they were able to learn something about X inactivation and for their proposals. So what they found was, of course, in females you have 1 inactive X chromosome, and in males you have none. However, in triploid X, there are 2 inactive X chromosomes, and Klinefelter male patients have 1 inactive X chromosome. So because male patients, although they are Klinefelter's patients, have 1 inactive X chromosome, this really tells you that the presence of a bar body isn't strictly to do with the gender of the individual or the sex of the individual. Finally, in the tetraploid female cells, there are 2 inactive X chromosomes. And so part of Mary Lyon's hypothesis was then that only 1 X chromosome remains active per diploid set of autosomes and all others are silenced. So in the case of trisomy X, you can see that 2 need to be inactivated so that you only have 1 remaining active for those diploid set of autosomes. Whereas in the tetraploid female cells, because you have not just a diploid set of autosomes, but in fact twice as many, now you need 2 active X's for that, and therefore you have 2 that are being inactivated. And as I mentioned, in Klinefelter's, you have 1 active X chromosome and 1 inactive X chromosome. And so it's not dependent on that, whether or not a Y chromosome is present in the cell, or indeed whether the patient is male or female, but rather on the how many X chromosomes should remain active within each nucleus. What's quite interesting is that Mary Lyon made her hypothesis in 1961, but relatively quickly, within 5 years, there'd already been a change, at least in terms of how you would test for sex within the Olympics. So in the 1966 Olympics, they actually used some tests to look at whether or not athletes had a bar body to be able to determine whether or not they were truly female. Now, this is rather interesting because, of course, given what I've just told you, the Klinefelter's male patients, were there any to be in the Olympics, would also come up as female. But this is not the way around they were concerned about. They were really concerned that males were, of course, competing as females. But it's interesting, it really changed how Olympic testing happened within 5 years. So other than how many X chromosomes will be inactivated, the second part of Mary Lyon's hypothesis is the random nature of X inactivation. So we've discussed this before, thinking about the calico cat, and I'd like to come back to it again because it's really a nice and an important finding that she had. So she wasn't actually studying calico cats, she was studying something very similar, the coat colours in mice, an experimental model system. And what she found was that if you looked at variegated phenotypes, and what this means is phenotypes of the coat colour that have multiple different, or at least 2 different coat colour phenotypes, or in other cases other phenotypes. So you can have variegated ivy or variegated other plants, and they have 2 different options of colours within their leaf. But cells within the same tissue can have mosaic expression, so they can have different outcomes. So different colours for variegated ivy, different coat colours in the case of mice or cats, for example. And she noticed that if you had females that were obligate heterozygotes, you had to know that they were a heterozygote for a particular coat colour allele, then they could have this mosaic or variegated coat colour. But males never showed this variegated coat colour. And of course, the same effect is seen in humans if you're able to see this within the skin colours, and the same thing is seen in cats. And I would like to go through that cat example, the calico cat example again in more detail. So this is really about the visual appearance of random X inactivation. So if you consider that there are 2 alleles of the ginger gene, you have the ginger gene itself, which is here given as a big G, and then you have the black version of this. It's a null allele, and it would be little g. Well, normally, ginger would be dominant over the black allele, and therefore the cat would end up being ginger. However, if now we think that we know there's X inactivation, we know that half of the time, then the ginger allele will be on the active X chromosome, as shown on the left-hand side here, and so that cell will end up producing ginger pigment. However, half of the time, for the rest of the time, you'll have the ginger allele rather than the black allele being on the inactive X chromosome, and this will result in black pigment being produced. So this choice, when it's set up, happens at around gastrulation and is then mitotically heritable for the rest of the lifetime of that female cat. And so you end up getting these clonal patches of coat colour as shown in the picture of the cats on the right. And the same is true if you look at a similar sort of a phenotype in mice, and this is what Mary Lyon was looking at. So as I've mentioned before, we know that we really don't see male cats that display this calico appearance. And if they are, the ones that are found, in fact, are karyotypically XXY. And so from what I've just told you, they're Klinefelter's cats, if you like, and they have one X chromosome that's inactivated, and that's why they display this similar phenotype to the females. So it was studies like this that led Mary Lyon to make her hypothesis in 1961. So the key point here is that random X inactivation occurs very, very early in embryogenesis, around gastrulation, and once that choice is made, it's mitotically heritable. It's also interesting to think, remember I mentioned the 1966 Olympics, they were already testing for the bar body to tell whether or not it was a female or a male athlete. But in 1965, Stanley Gartler also used this presence of the bar body, or random X inactivation, to prove that cancer was clonal in origin. So he could find that, in fact, if you looked at which X chromosome was inactivated, then all of the cancer had the same X chromosome inactivated in females. And this therefore suggested that they all were derived from the same single cell. And so you can see within a very short space of time of X inactivation being proposed, it had already infiltrated other fields within biology. So in the next lecture, we'll think about the different types of X inactivations that occur in mammals.